our first speaker knows everything about life, I would say. Why don't you come up to me, Professor Hans Clevers? May I say Hans to you?
Yeah.
Good afternoon. How are you? So, you do something that is total magic to me as a lighting designer. You go in the human body with light, is that right?
Well, we go into the mouse body and we create mice that emit light. That's what we do.
Well, that sounds like a new way to distribute light in this world, right? But you are on the hunt for something else, I understand. Can you very quickly tell me?
Yeah, very quickly. So what we use light rather than that we study light. We use different colors of light to study processes in live organisms. So we can engineer, I'll give examples, we can engineer animals to emit light in ways that we design on a computer. And then we can basically follow those mice, whether alive or jellyfish or whatever you do this to, and study the processes by just looking at the light that these animals emit.
Well, you sound like a person who can talk about it for days. You've got 15 minutes with this audience. This white spot is all for you. I hand you the clicker. Good luck in the year of light.
Thanks. Okay, so I changed my title slightly, so it is not about creating humans that emit light, it's about creating mostly mice that emit light. On my first slide, you can see a problem that all of us sooner or later, and I see a few good examples in the audience, will encounter, which is aging. And aging, in essence, is due to the fact that we are built of tissues, and tissues are built of cells, and cells work very hard, and like anything that works hard, the cells get worn out, and they have to be replaced, which is not simple. I'll say a lot about that. It not only happens to the skin and to our hair, which is quite obvious, but I actually found a very good other example on the internet that you probably recognize. It, for instance, also happens to our muscles. And any other organ in our body will actually work hard and cells wear out and get replaced. So we're not defenseless against this process. For about maybe 80 to 90 years, we have a defense mechanism that's called our stem cells. And every tissue we assume or we know now has its own stem cells. And what stem cells do, they're rare, they're small, they're difficult to find. Every organ has its own. When new cells are needed in an organ, when they need to be replaced, a stem cell basically copies the DNA. Every cell has the entire information of my entire body in my, as I stand here. And those daughter cells will then perform the functions of the organ where they sit in this case it's a liver, but also there's always in this process another stem cells is set aside. So if there's damage a year later, an evening of heavy drinking actually does this, stem cells get active and replaced the cells that are lost. About maybe 15 years ago, my lab became interested in studying stem cells, trying to identify stem cells. Very few were known at that time, and we realized that the organ that you see here, the gut, was probably the best system to actually study how stem cells work and learn how to find them, prove that they are stem cells, and use them in the lab and maybe later on in a hospital. Here you look on the insides of the gut. These extensions are called villi, or darmvlokken in Dutch. They help us digest food and take up nutrients efficiently. These cells are constantly replaced by cells that are produced in these structures that are called crypts, the Crypts of Liver. Every medical student learns this. And what we realized is that when one thinks about the process of how this cell renewal occurs, you see a villus, a darmvlok in the middle, surrounded by these little holes called crypts. The holes produce new cells, many new cells every day, and they actually have a very short half-life. They only live for 4 days. They get born, they divide rapidly, they perform a function while they move up the sides of the villus here, and 4 days after they're born here, they will die at the tips of the villi. So the entire organ is replaced every 4 days. It's about 200 grams of cells that you make per day in your gut. If I would do that with all of my organs, I would have to produce 20 kilos, 25 kilos of tissue every day. So incredible production of cells. So we thought this is probably the best and the fastest system to find out how to deal with stem cells. We started reading, this is now 2006, where our textbooks tell us this is a crypt. In light blue, you see the rapidly proliferating daughter cells. In yellow, a cell type that's called a Paneth cell. And somewhere hidden in that mass of cells, there should be rare, small stem cells. A British postdoc in my lab, who is now in Singapore, started working on this, and he found a molecular flag. Now, I won't try to tire you with molecular details here, but in biology, it's very nice if you have a molecular marker, a flag, sitting on one cell but not on the other, because then you actually have a handle to start studying those cells. Now, we found one with the prosaic name LGR5, a gene that was never studied before, and with that marker in hand and these little brown stripes were totally unexpected to us. They are cells, every stripe is a cell that we had entirely missed. And they were in the exact position to be the stem cells. So we're very happy. We then realized that we were not the first to see these cells. There was a guy, Leblanc, who passed away actually in the year that we published the paper. And we found papers from him from the 60s. He actually worked with the Curies. He didn't study light, but he studied radioactivity, but he used radioactivity in human and mouse bodies. A fantastic man with a fantastic biography. Still active at the age of 2004. I gave his first PowerPoint presentation and remarked, a month ago I thought PowerPoint was a tool for sharpening pencils. So he had actually postulated, everything I'll show you, he had postulated in these beautiful papers from the 60s, all by just looking at sections of tissues and of mice and of humans, but he could never prove what he was seeing, because he didn't have the tools, the light tools that I'll show you, that we now have in our hands. Now, what can we do? In the Hubrecht Institute, we can actually call up the entire genome, the entire genetic code of a mouse on a computer screen. Here you see a chromosome, one of the 20 or so chromosomes that a mouse has, and then we can design a change in that code, and then we can create what we call a knockout or a knock-in mouse that has that change in the code. And then it takes about a year, you have a mouse that actually has added information or deleted information, and will hopefully do something that you designed on that computer. The mouse, when it actually produces offspring, will hand over that design also to its offspring. So you really make a new version of the mouse. Now, what did he build in? Two things. He stole a genetic trait from small animals, fireflies, jellyfish, animals that can emit light. They produce fluorescent proteins. So a small piece of DNA was sent to us in the mail by a lab in California. Nick put it in the mouse DNA and made a mouse in the hopes that the cells of Leblanc, these little brown stripes that we can only see in that tissue with that particular technology, would now emit light in a live mouse. And then, so that's called green fluorescent protein. And then he put in another trait, another piece of DNA that we stole from a virus, and I won't say too much about it, but it allows us to shuffle DNA in mature in an adult mouse at will. So the green cells will have an additional capacity, namely when we tell those green cells by injecting something into the mouse, reshuffle DNA, they will do so. And I'll get back to that. So this works well. So here you see 3 of those darmvlokken, of those villi, live tissue now. So this is no longer fixed and under the microscope. This is in a live mouse. A number of crypts, and the cells of Leblanc emit, you see these little green stripes, emit green light. So they are like the fireflies where we stole the DNA from. We then crossed this mouse, so with the trait that the cells are green and can reshuffle their DNA, with the piece of DNA that can be reshuffled. So we made another mouse that actually encodes 4 different colors. You see green, yellow, red, and blue, and if you look carefully, green and yellow are in inverted pairs, and red and blue. This mouse has no color. However, if we tell it to reshuffle the DNA, it will stochastically end up with one of these 4 situations. So the on-off switch sits in front, that's this thing here. So this mouse is off. If we remove this, it's on, but then it can reshuffle this and become green, yellow, red, or blue. And biologists here might know that we have 2 chromosomes, two copies of every chromosome. So actually when you have this mouse homozygous, it makes in a checkerboard 10 false colors. So you can make a mouse now that can emit 10 different colors. And the idea would be, and this is the impression of our animator, that we could give all of the stem cells a different color, all of the cells that we hoped are stem cells. Over time they will produce offspring, and slowly the crypt will fill, in 2 days or so, will fill with colored daughter cells. And in this particular case, the red stem cell is winning. It's apparently the best one of the 15 or so that sit there. And then our animator had a vision that this is what that mouse should look like on the inside. Now, we all agreed this was a little over the top. But then we got the real results, and these are the guts of live mice, where we go inside with the machinery, the equipment that allows us to visualize this. And then on a blow-up, what you see here is, in essence, are 4 or 5 of those crypts. The stem cells will produce light in their own individual colors in live tissue, and essentially the gut of this mouse looks like, that's why we call these mice the confetti mice. They're entirely full of different colors. We then found out that many other organs have green cells. Those organs also pick up colors, and even the skin, the hairs have different colors. And we had a test where we let one mouse escape, and then with the black light we could find it because its skin looked the same as what you see here. So this worked, and this actually proved that the cells are the stem cells. You mark a stem cell, it makes a lot of daughter cells, and for the rest of the life of that mouse, the gut looks like this. So every day these stem cells crank out more colored daughter cells. So now we have a mouse where we have proven that the green cells are the stem cells. We can now take small biopsies, small bits of tissue from a live mouse. It's often done to patients. Mice don't really care so much. And this is one bit of tissue that you see here on the left, one crypt. The green stripes, each stripe again is a green stem cell. We can now isolate pure live stem cells, and we know that they are stem cells because they are green. Every dot there is a green stem cell, and it's alive. And with this in hand, a Japanese postdoc in my lab, now back in Tokyo, Toshiko Sato, decided if he could mimic this process that occurs in vivo, if he could mimic it in the lab, the fact that stem cells divide and make more cells, and do that for a very long period of time. This was against dogma, and no scientist would try this with a sane mind, because all the textbooks say normal cells do not grow outside the body. Cancer cells will, but normal cells will not. This is what Toshi actually got. Single cells produce a lot of pretty chaotic tissue originally, but then when you break it up, you might appreciate that these are, maybe you need a little bit of a trained eye, but they're quite regular. And now again, if we do this from a mouse where the nuclei emit a green light, and we make movies over time, what we essentially see is that we produce from one stem cell a mini gut, a mini darm, in the lab, and we can make billions of them from one stem cell, and we can actually put them together and they make a tube. There's a central lumen, that's where the food would be, and then there are multiple crypts. The stem cells sit here, as you see in the back. Essentially, this single cell knows it comes from the gut, and it makes a gut when you put it in culture. It was extremely surprising. Now, how normal are these things? A final experiment, and then I'll finish. Toshi actually grew mini guts from one stem cell from a mouse that was entirely red. So these mice, they glow in the dark, they're red. Every cell is red. You can take a stem cell out of the colon, out of the large intestine. He grew it up to about 1 billion cells, froze them, sent them to Tokyo by FedEx, and apparently the most difficult part of the project was to get this sample past customs, he told me, in Japan. But then when they arrived in the lab of his former PhD advisor, they injected these organoids, so the red Dutch organoids, into mice, Japanese mice with a black gut that had colitis, so they had defects in their distal colon, very common disease in humans as well. And you can see these ulcers, the big holes that sit in the lining of the gut. This is the colitis. And apparently these mini-guts, these Dutch mini-guts, still know where they come from. They not only build a mini-gut, but they will go to these lesions, they will adhere, and they will open up. And like a living Band-Aid, they cure these mice. So a single cell was used to cure about 40 mice in Japan. And the single cells were grown in Holland. Now, some of you might have realized that this was not the real experiment that is here. So what you see here is black Japanese tissue, Dutch epithelium, Dutch gut cells that were grown in the lab by Toshi. And by any other technology, you cannot see where the Dutch tissue stops and the Japanese tissue starts. So it's fully integrated, it's fully functional, and these mice have now lived 1.5 years since their transplantation, and they are still doing very well, this Dutch tissue. The promise of this is now that we can do this for human organs, and actually we're publishing today 2 papers on this subject. Rather than transplant a liver from a dead traffic victim to a single patient, take a small biopsy from a live volunteer, expand it in culture to as much tissue as we want, freeze it, and then when somebody needs a transplantation, that's the promise, we're not there yet, we can just get the right tube out of the freezer and give it to the patient on a planned day, rather than in these horrible circumstances surrounding traffic accidents and dead donors. With that, I think I just want to name a few people. The most important person in my lab was Nick Barker, who struggled forever to find this stem cell for like 7 or 8 years, and then all of a sudden found this marker, LGR5. And Toshi Sato, the Japanese postdoc, who against all advice and all dogmas decided that it should be possible to culture stem cells. And of course, we thank Light for the tools that it gives us to do these experiments. Thanks very much for your attention.